Preparing for Lab 6, How to Grow a Culture. In Lab 6 Part A, students first concentrate the nice E. coli cells that have produced the RFP protein. In Part B of Lab 6, students then purify RFP using hydrophobic interaction chromatography. Depending on your distribution center, you may have different options for how to complete Lab 6. The first option is receiving a prepared Lab 6 culture. This allows you to immediately begin Lab 6 Part A with your students. The second option is to grow your own culture in class, which you will then use to complete Lab 6. The final option is to receive already prepared supernatant. Supernatant is the fluid containing RFP that has been extracted from the bacterial cells. If you elect to receive supernatant, you would start Lab 6 at Part B on Step 9, where your students add binding buffer to 200 microliters of supernatant. This is a great option if you feel rushed for time. Today, we will be reviewing how to grow your own culture for Lab 6. Growing a Lab 6 culture entails growing a culture of transformed E. coli cells and ABE-LA broth to the mid-log phase of bacterial population growth, and then initiating the production of RFP by adding arabinose to the culture and letting the culture grow for 24 to 48 hours. How does RFP expression work? The Lab 6 cells are transformed cells that contain the P-ARA-R plasmid. Recall that the P-ARA-R plasmid contains the RFP gene along with other important genes that allow for the bacteria to produce red fluorescent protein. The scientists that designed the ABE labs placed the RFP gene under control of the arabinose operon. An operon is a unit made up of linked genes which regulates other genes responsible for protein synthesis. The arabinose operon usually controls the expression of 3 enzyme-coding genes: ARA-B, ARA-A, and ARA-D. These 3 genes encode enzymes that digest arabinose sugars and are only expressed when arabinose is present. Hence, arabinose is like an on switch for the expression of the genes controlled by the arabinose operon. For the ABE labs, the enzyme-coding genes were cut out and replaced by RFP, which places the RFP gene under the control of the arabinose operon. The arabinose operon is turned off by the expression of the ARA-C gene. The ARA-C gene produces the protein ARA-C, which binds to the promoter region of the RFP gene, causing the DNA to form a loop conformation that blocks the RFP gene from being transcribed to RNA by RNA polymerase. Because RFP expression is controlled by the arabinose operon, RFP expression only occurs in the presence of arabinose. When arabinose is present, it binds to the ARA-C protein, which changes its conformation and causes it to unbind from the RFP promoter. With ARA-C no longer attached to the promoter region, the DNA loop is released. The ARA-C arabinose complex then helps stabilize RNA polymerase, allowing for the RFP gene to be transcribed and then translated to the RFP protein. When to add arabinose? Understanding the bacterial growth curve. Bacterial population growth follows a known pattern called the population growth curve of bacteria. This pattern can be visualized with the graph that shows the log of bacterial population size on the y-axis and time on the x-axis. After inoculating your ABE-LA broth with Lab 6 bacterial cells, the bacteria enter what is known as the lag phase of the population growth curve. During the lag phase, bacterial population growth is stagnant. The cells are becoming acclimated to their new environment and begin to synthesize the enzymes they need to initiate cellular division. The individual cells grow in size, but cellular division has not started or is just beginning. After some time, cellular division begins and the bacterial population grows rapidly. This occurs during the log phase. During the log phase, bacterial growth is exponential, meaning the rate of new cell production becomes faster over time. The bacterial population is the most resilient during the log phase, as there are plenty of nutrients available to support cellular growth, cellular division, and the production of proteins. As the population grows, nutrients become depleted and cellular waste becomes more prolific, causing population growth to slow. Eventually, the rate of cellular division equals the rate of cell death. This is called the stationary phase. Eventually, the rate of cell death may exceed the rate of cellular division, resulting in a dip in population size. Arabinose is added during the mid-log phase of bacterial growth to maximize the amount of RFP produced by bacteria in our culture. The production of RFP takes energy. Hence, if you induce the production of RFP too early, the bacteria will have less energy to devote to replication and population growth of the bacteria will be slowed. It may take significantly more time to obtain a culture with a high concentration of bacteria, and hence a lot of RFP, if RFP is added too early to your culture. If you induce the production of RFP too late, near the end of the log phase or beginning of the stationary phase, the bacteria will be under more stress as nutrient sources are depleted and toxic cellular waste begins to accumulate. This reduces the capacity for the cells to produce RFP, meaning your culture will not contain enough proteins to purify using HIC. You can visualize when arabinose should be added using this image of McFarland tubes. McFarland tubes are a visual representation of what different concentrations of bacteria look like. During the log phase of bacterial growth, you will want to add arabinose to your culture when the turbidity of your culture matches that of the McFarland standard numbers 3 or, at the latest, 4. Now that you understand how RFP is produced and the role bacterial growth curves play in RFP production, let's go through the steps of growing a culture. To grow your own culture, you will need a shaking incubator, a Lab 6 culture kit, which contains a sterile 125 milliliter flask, falcon tube of LB ampicillin broth, and a vial of arabinose. You will also need a vial of Lab 6 cells, a P1000 pipette, P1000 pipette tips, and a biohazard bag. Optional materials include a white index card, Sharpie, scissors, and tape. Note that the LB broth and arabinose should be stored in the refrigerator and the Lab 6 cells stored in an isofreeze unit in your freezer until you are ready to use them. Start by opening the packaging of the sterile flask. An optional step is to make a McFarland tube-inspired background for your flask. You can do this by cutting an index card in half and drawing straight lines on it using a black marker. You can then tape the index card to the back of the flask with the lines against the flask. This creates a contrasted background for your culture. As the bacteria replicate, the LB broth in the flask will become cloudy, and the black lines on the index card will become harder to see. This makes it easier to visualize bacterial growth so that you know when you have reached the mid-log phase. The next step is to open the falcon tube of LB amp broth and pour it into your sterile 125 milliliter flask. The vial of arabinose may be taped to the LB amp broth tube or packaged separately. The arabinose should be set aside in a tube rack as it will not be used for a few hours. Once you pour the LB-AMP broth into the sterile flask, use the P1000 pipette to transfer the vial of Lab 6 cells into the sterile flask. Be sure to dispose of the empty Lab 6 vial and pipette tip into a biohazard or autoclave bag. Then close the cap of the flask and give it a little swirl to mix in the Lab 6 cells. Place the flask into one of the flask clamps on the shaking incubator. The flask should be snug, not loose. Turn on the unit by pressing the power button. Then, adjust the set temperature by using the up and down arrows. Cultures are grown at 37 degrees Celsius or 98.6 degrees Fahrenheit, which is body temperature. But note that the temperature settings of some shaking incubators can be off by a few degrees. Hence, please follow any notes like the one on this shaking incubator, which indicates to set the temperature to 39 degrees. Press the on button next to the arrows to turn on the heat. You should see the display temperature gradually reach your set temperature. Next, turn on the shaker table. Start at a low rotation and gradually increase to the highest setting you can reach without the broth splashing onto the cap of the flask. Do not turn on the timer. You want the shaker to remain on continuously. You will need to let the culture shake for between 2 to 3 hours until you reach the mid-log phase of the bacterial growth curve. You can tell you have reached the mid-log phase when your culture becomes cloudy and the striped background becomes harder to see. After about 90 minutes, you should check the culture every 15 to 20 minutes to see whether it has reached the mid-log phase. When you first start, the LB broth will appear yellowish but clear. After about 1.5 hours, the broth should begin to become cloudy, indicating that the bacteria are replicating and that the concentration of bacteria in the culture is increasing. After about 2 to 3 hours, the culture will become cloudy such that the striped background on your flask is hard to see. This is the mid-log phase and is when you should add arabinose to initiate the production of RFP. Once you reach the mid-log phase, turn off the shaker table and remove the culture flask. Remove the cap of the flask and then use a P1000 pipette to add the arabinose solution to your culture. Tightly recap your culture, give it a gentle swirl to ensure the arabinose is in the culture and not on the side of the flask. Then, place the flask back into a tightly secured flask clamp. Turn the shaker table back on, as well as the heat if you turned off the entire unit, and let the culture grow for 24 hours. After 24 hours, your culture should appear hot pink, indicating the bacteria have produced a lot of RFP. If your culture looks salmon colored, let the culture shake for an additional 24 hours until it appears hot pink. If for any reason your culture fails to turn pink, please contact your site coordinator immediately. They may be able to provide you a culture to complete Lab 6 or provide you supernatant so that you can still at least complete the HIC portion of the lab with your students. More videos on Lab 6 are available on the Amgen Biotech Experience LA channel and the Amgen Biotech Experience channel. If you have any questions, please reach out to your site coordinator.